Okay, so ready to talk about the integumentary system. So we're finally at a point now where we're going to just kind of go system by system the rest of the semester. Mentioned, we in the very first lecture we talked about those levels of organization. We kind of skipped the first level. We didn't really talk too much about the chemical level where you got atoms and molecules and bonds. So we're jumping ahead now. From there, we talked about the cellular level, then we talked tissue level. Now we're going to talk about organs and organ systems the rest of the semester. Our first system here is the integumentary system. And our first organ that we'll talk about the most in this lecture is our skin. And stuff you're filling out right now. As a whole, other organs of the integumentary system include, aside from the skin, that is, the accessory structures of our hair, finger and toenails, glands like the sweat glands, oil glands that you find in the skin, various blood vessels that are in the skin, same with muscles and nerves, okay? But mainly in this lecture, we're going to talk about the skin, your hair, your nails, and the integumentary glands. Okay. Overall, the organs of the integumentary system, and mainly we're talking skin here, provide us with numerous functions. That includes maintaining the body's integrity, maintaining temperature, converting inactive vitamin D to its active form, providing sensory information, and telling us when we're hot, when we're cold, when we're itchy, when we touch something hot or painful, something like that. And then just overall just helps maintain homeostasis or balance in our internal environment. Okay. So like I said, we'll start with the skin. Other terms for skin, we learned this in our previous lectures: cutaneous membrane. Another term for it is integument, which is probably where that integumentary term comes from. I'll just call it skin, okay? But it consists of different tissues joined to perform specific activities. It covers our external surface of the body and is our largest organ of the body in both surface area. And that is about 2 square meters or 22 square feet. And it's also the heaviest. It weighs the most, about 4.5 to 5 kilos, or 10 to 11 pounds, which is around 16% of your total body weight. Now, obviously, that's different from person to person, so that's just an average. If you're, a really large individual, you're going to have more skin. If you're a small individual, you're going to have less skin. Thickness of the skin ranges from 0.5 millimeter or 0.02 inches thick in our eyelids to 4.0 millimeters or 0.16 inches thick on our heels, the bottom of our feet. Most of the body, though, you're going to find it is about 1 to 2 millimeters thick. So, it's really not super, super thick in most places. 1 to 2 millimeters is not a whole lot. It's still very protective, but it's not completely impenetrable. Something as simple as a piece of paper can cut through it. So it is strong, but not the strongest thing on earth. I'm sure you all know what dermatologists are: a doctor that performs or practices this type of medicine, dermatology, the medical specialty dealing with diagnosing and treating skin disorders. Now, there are two principal parts to the skin: the outer thin epidermis, which is made up of epithelial tissue. We'll kind of start talking about that here on the next slide and next couple slides. And then deep to that is the much thicker dermis, made up of connective tissue. But then just deep to the skin is a layer, and it's not technically part of the skin, but it's, as you can see, connected to it. It's made up of areolar and adipose tissues, which we talked about those in our last lecture. We call this the subcutaneous layer or hypodermis. Now, the word cutaneous, we just learned, means skin. The prefix sub is just kind of a fancy way for just like deep or below. Okay, like a submarine is a vessel that spends most of its time underneath the the surface of the water. And kind of the same situation for the prefix hypo. Hypo means low or below, and in this case, below the dermis. Okay. Now, looking at the illustration down here at the bottom, I'm going to tell you right now, this image will be on the exam next week. But with that said, I'm not going to ask you to know everything. Okay. So what I'll do is I'll tell you what to highlight or star, and these are going to be the things that I want you to know. So anything else, don't worry about. And if you haven't already worked on your labeling packet, which will be due the night before the exam, this picture is in there. When you go through and label it, don't worry about labeling the things I'm not going to tell you to study for the exam. So that don't waste your time on that. Just label the structures that I want you to know. So With that said, we'll start here. I'm just going to work on the left side, going from top to bottom. So the things that I list off here are the things I want you to know. Okay, so we'll start with epidermal pegs, dermal papillae, sebaceous gland, erector muscle of the hair, which we'll kind of have another term for that, same thing. Hair follicle and hair root, eccrine sweat gland, apocrine sweat gland, and that's it on the left side. On top, I want you to know the hair shaft. The right side, I want you to highlight where it says epidermis. And then the two parts of the dermis, papillary and reticular, and just the dermis as a whole. And then subcutaneous layer, or tissue, I should say. So that is the structures I want you to know. Okay. Anything else? Don't worry about it. Don't label them in your labeling pack or anything like that. All right. Like I said, we're going to start by talking about the epidermis. And then we'll talk dermis here in a couple of slides once we've gotten through that. First thing I want to talk about with the epidermis are the different types of cells that make it up. There's 4 principal types of cells: keratinocytes, melanocytes, you've got Langerhans or Langerhans cells, also go by the more fancier term intraepidermal macrophages. And then Merkel cells, which also go by the fancier term tactile epithelial cells. And then there's a 5th cell that is listed here. It's not kind of a type of cell as far as specialty. It's just an undifferentiated cell that we'll talk about just briefly, but it's not a picture at the bottom. So looking through and talking about them, first of all, just to review, we talked about this the other day, but your epidermis is made up of keratinized stratified squamous epithelium, which we learned stratified has multiple layers. Squamous tells you that the cells are flattened or plate-like, and cells have keratin in them. Okay, the cells that have the keratin are literally called the keratinocytes. Most numerous of the cells, 90% of the cells, the epidermis, and so it's very, very much so the majority of the cells. We talked a little bit about keratin the other day, but it's a tough fibrous protein, provides protection from things like heat and microbes and various chemicals, and even helps with waterproofing the skin so water doesn't, just leak through either into or out of the body. Okay, we arrange our keratinocytes in 4 or 5 layers. We're going to talk about the layers of the epidermis on the next page, and that's all just dependent upon the thickness of the skin. Like we talked about, your eyelids is thin, your heels is really thick. Keratinocytes also produce what we call lamellar or lamellated granules. These are going to release a water repellent sealant. It's kind of a fatty carbohydrate thing that decreases water entry and loss and inhibits the entry of foreign material. So that also helps with waterproofing along with that keratin. Now, when the keratinocytes produce the keratin, it accumulates inside. And what's constantly happening, I'm just going to kind of jump ahead to the next slide. In the very deepest part of the epidermis, we're producing new keratinocytes through mitosis. And eventually the cells that are produced here, and we can actually see an example right there where 2 cells are being formed from the original one. Those cells get pushed outward, and they continually get pushed outward as new cells are being formed. And eventually the cells make their way to the very outer surface of the body. And by the time those cells reach that point where they're on the outside of our body, they're just dead scale-like structures full of keratin. So the cells that we're scraping off, sloughing off all the time are just dead cells. It's kind of gross, but they say a lot of the dust that collects in your house and your place of living is dead skin cells. So we're constantly doing that, and that's just fine because we're constantly replacing them with new living keratinocytes. Okay, the next type of cells are called melanocytes. They represent 8% of the cells, and they protect the skin against damage from UV radiation. Ultraviolet radiation comes from the sun, most notably, in its natural state. You can also get UV rays from like going into a tanning bed, which is very unhealthy. It's really bad for your skin. So if you do it, just be careful. Tanning's not a great thing. Okay, what melanocytes are? They're very cool. They're spider-shaped cells with long, slender projections extending between the keratinocytes that allows them to transfer melanin granules to them. So looking at the picture at the bottom, kind of have these little spider leg-like structures. Okay, they produce this melanin, and these leg-like extensions kind of surround the keratinocytes, and then those melanin granules are going to be transferred to keratin. So what is melanin? It's a yellow, red, or brown black pigment that contributes to your skin color and absorbs damaging UV light. So in other words, whether you're light skinned, dark skinned, whether you tan very well is all dependent upon the type of melanin that you produce and how much. Okay, and most of that relates to your genetics. Okay, now the melanin is very important because it's going to basically come into the keratinocytes and create like an umbrella that protects the nucleus that's inside of those keratinocytes. And that umbrella, like a natural umbrella, can provide shade. It protects from that UV radiation that's going to come down from the sun or from the tanning bed. And what UV rays can do, that radiation can damage the DNA inside. So by transferring that melanin over, it protects those cells from that DNA damage. Okay. Unfortunately, melanocytes themselves are particularly susceptible to damage by UV light. And if you've ever heard of the term melanoma, that's the most dangerous form of skin cancer. They call it melanoma because guess what type of cell it originates in? Melanocytes. Okay. All right. The third type of cells are called Langerhans cells. That's the kind of traditional term. Probably named from some scientist or doctor that discovered them. They're kind of pushing this newer, fancier term for them, intraepidermal macrophages. I want you to know both of their terms, but they're involved in immune responses. These guys are more like a white blood cell. In fact, they actually arise in the same place that our white blood cells do. Our red bone marrow, the bone marrow that we'll talk more about in our next unit. They then migrate to the epidermis. Okay. Their role in the immune response is to help other cells of the immune system recognize an invading microbe and destroy it. So they don't actually do the destroying, but they can like tell the immune cells, like your other white blood cells, say, hey, this virus is in here, this bacteria is in here, come clean it up, come destroy it. Okay, so they're kind of like a tattletale. Merkel cells, which go by another fancier term, tactile epithelial cells, are going to function in sensation of touch. Hence the name tactile. Touch is a kind of fancier word, or tactile is a kind of fancier term for touch. They're always associated with a flattened structure called a tactile disc, which is the beginning of a sensory neuron. The type of cell that sensory neuron is going to transmit a nerve signal into the spinal cord in the brain. And so by stimulating these, you stimulate the neurons because they communicate with one another, and that allows us to feel when someone touches us or when something's touching our skin. They're the least common type of epidermal cell. We only find them at the junction between the epidermis and the dermis, so the very deepest part of the epidermis. Okay. And then finally, like I said, there's not a picture of these, but there are also very important stem cells found in the deepest layers of the epidermis. These are undifferentiated, meaning that they are not fancy like these. They haven't developed to this point yet. Essentially, what they're going to end up becoming, though, are the keratinocytes more than likely, since the keratinocytes are the most numerous of the cells. As far as those 4 pictures go, I only included them because they're in your textbook. Not going to ask you to worry about them for the exam. Okay, let's talk about the layers of the epidermis. Okay. Several distinctive layers of keratinocytes in various stages of development form our epidermis. In most regions of the body, there's 4 layers. So 1, 2, 3, 4, 5 are listed here. One of these layers is not found in thin skin. Okay. Now, in cases where we expose the skin to friction, like in our fingertips, the palms, which are like the inside of our hands, the soles, which are the bottom of our feet, the epidermis has 5 layers. We call this thick skin, and the thicker the skin is, the more protective it is, for hopefully obvious reasons. Okay, we're going to start by talking about the most superficial layer, which is the corneum, and then we're going to work all the way to the deepest. So if you remember, the term superficial means closest to the surface of the skin. Deepest is the most inside, which is the basale. Okay. All right. The once again most superficial layer is the corneum. It is typically 25 to 30 layers of dead scaly keratinized cells, ranging in thickness from just a few cells in thin skin to more than 50 or more in our thick skin. Okay. The interior of the cells here are full of keratin. Between the cells are lamellar granules that help to waterproof it. The cells within this layer, or within these layers, help overlap one another, like scales on the skin of a snake, or if you think like shingles on a roof, which is kind of cool. This is a layer that we've already kind of talked about how we're constantly flaking it off, scraping it off. Skin flakes and dandruff will be remnants of this layer. If you constantly expose your skin to friction, it will help form what's called a callus, which is described as an abnormal thickening in the corneum. And I say abnormal, it's not, to me, it's a normal thing. It's a protective thing, very common in people that use their hands a lot. So, on campus, I have a lot of softball players, baseball players, swinging a bat all the time. They're going to get those calluses. If you'll lift weights, that kind of stuff, you'll get calluses. They're not the most beautiful thing on earth. People don't really seem to like how they look, so they kind of will scrape them off or file them off. But your body's trying to protect itself by growing them, so keep that in mind. All right, deep to the corneum is the lucidum, a layer of 4 to 6 layers of flattened clear dead keratinocytes with lots of keratin and thickened plasma membranes. The cells here have no nucleus, which causes it to appear as a thin translucent layer above the granulosum. And that's why it's called the lucidum. Lucid means like clear. If you see lucidly, you're seeing clearly. Okay. This is something I would highlight right here, this last sentence. Very important. This is the layer that's only found in thick skin. So this, in other words, is not found in thin skin. This is that variable layer. If you're talking about thin skin, it's not going to have the lucidum. Deep to it is the kind of grainy appearing layer, and that's why we call it the granulosum. Okay, if you look in our picture here, you can kind of see the little red cells with the little black speckles. Okay, the granulosum is going to consist of 3 to 5 layers of flat keratinocytes. This is where the cells start to die off as they get pushed outward. We call this process apoptosis, a fun word to say. This is an orderly genetically programmed cell death in which the nucleus of the cell fragments before it even dies. And that's why the lucidum and the corneum, you don't see the nucleus inside. So apoptosis is not like an accidental death. It's a purposeful death. Okay. The cells that are going to eventually be lost off the skin don't need to be living until that point. Okay. This middle layer, the granulosum, is where that dying takes place. Now, a distinctive feature of this layer is the presence of darkly staining granules of a protein called keratohyalin, which converts tonal filaments into keratin. So this is where we form the keratin. I'm not going to really explain what tonal filament is. Tonal filaments are keratohyalin, but just know this is where we form the keratin. Lamellar or lamellated granules here secrete that glycolipid that waterproofs the skin that I've been talking about. And this is going to help form a barrier between the surface cells and the deeper layers, cuts the nutrient supply off to those surface layers, and slows water loss across the epidermis. So it's basically a barrier between the deeper layers, which are all living, and the outer layers, which are not living. There's no reason to have nutrients, diffusing out to the lucidum and the corneum because the cells aren't living anymore. There's no reason to, provide them with any nutrients. The spinosum is the 4th layer. It consists of 8 to 10 layers of many-sided keratinocytes fitting closely together. It's called the spinosome, and I kind of pronounced it that way on purpose because it's kind of a spiny looking layer, and that's because of the spiny projection of the keratinocytes forming tight junctions, providing both great strength and flexibility to the skin. Okay, it's kind of hard to see, but there are these little connecting things between the cells. We talked about tight junctions. Those were something we discussed just on Tuesday. Okay. This layer is the first layer where we're going to see any other type of cell aside from keratinocytes. We'll see those Langerhans or intraepidermal macrophages. Once again, those are going to help provide that immune response. And we even see projections of melanocytes. And then that brings us to the final layer, the basale, which also goes by the term germinatavum, but I want you to know mainly as basale, and that's what I call it. It's a single layer of cuboidal or columnar keratinocytes sitting on top of a basement membrane. Some cells in this layer are stem cells undergoing mitosis to form those new keratinocytes because this layer produces the cells for the other layers due to the upward movement of these cells towards the surface. The other name, germinatavum, refers to the fact that this is where things are germinating or sprouting. In other words, this is where we're forming the new cells. This is also the layer we're going to find those melanocytes that produce the melanin to give the pigment to those keratinocytes nearby. About 10 to 25% of the cells of this layer are those melanocytes. We also find those Merkel or tactile epithelial cells with their associated discs scattered throughout this layer for touch reception. And this layer is firmly attached to the underlying dermis along a wavy borderline. And we'll talk more about that here on the next slide and that connection that exists between the epidermis and dermis. Now, looking here at the bottom of the page, we've got essentially the same picture twice. This is the real deal, showing you what things look like under a microscope, zoomed in 240 times with the naked eye can see. You can kind of see the different layers that you can see. They're very nicely labeled, but it's really kind of harder to tell them apart there versus over here where they've kind of colorized things really nicely. So as far as the two images, this is the one I want you to know on the left. And it's just the 5 layers that are labeled on the left side of the picture. Don't worry about any of the cells or any of the other stuff that are labeled there kind of on the right side of the image. It's just the 5 layers I want you to know. Now, to help you guys, we'll see this, and I'll use these kind of a couple different points in the semester. You guys ever heard of a mnemonic device? Okay, where you can, use a phrase, and the first letter of each of those words in that phrase represents the first letter of a structure. And I looked some up the other day. I couldn't find any that I really liked, but we kind of came up with one when I was teaching this yesterday. So we'll use the phrase, if you want to write this down, it might help you, but come, so the C for come, C for corneum, let's, L for lucidum, get, G for granulosum, some for spinosum, bread for basale. And, I would say a student kind of came up with that better than me, but come, let's get some bread. That might help you remember those 5 layers in their correct order. If you wanted to use another mnemonic device, you could look for it online, but I'm not going to do that. Okay, let's move on from the epidermis now to discuss the dermis, the inner thicker layer made up of connective tissue, which is where a lot of the strength, flexibility, elasticity comes from for the skin. The very bottom of our text here, we're also going to talk about, just because we don't spend a whole lot on this lecture talking about it, but so I threw in the hypodermis or subcutaneous layer, a little information about it as well. And then at the bottom, you can see we've got that same picture that I already had you highlight, learn some things on. I did that, included it on this page just so I can kind of refer to some of them again. And then on the right, we've got two images showing you what the real deal looks like for the dermis and epidermis under microscopes. Kind of two different views. I'll kind of use this one a lot because it's really helpful. All right, reading through, the dermis is composed of a strong connective tissue containing both collagen and elastic fibers. This woven network of those collagen and elastic fibers provides great tensile strength, extensibility, or the ability to stretch, and elasticity, which means recoil to our skin. Okay, so your skin has the ability to stretch and recoil like a rubber band, unfortunately. And I kind of talked about this the other day. That is something that kind of diminishes as we get older, and that's why wrinkles develop, especially in older age. Dermis is much thicker, and you can see that very nicely in the picture at the bottom compared to the epidermis. We don't see nearly as many cells, but what cells we do see include fibroblasts in largest number, some macrophages, which are a special type of white blood cell, and then a few adipocytes or fat cells near the boundary of the subcutaneous layer. What we do find in this layer are lots of accessory structures. We see glands, we see blood vessels, and we see a lot of, and you can kind of see these yellow squiggly lines periodically, a lot of sensory receptors. Those are going to provide us with information like pain, pressure, temperature, like hot and cold, when you're itchy, when you're being tickled, so on and so on. So a lot of different sensations involve those receptors. Two layers make up the dermis, a thin papillary layer and a thicker reticular layer. The thin papillary layer is the superficial layer. It's made up of loose areolar connective tissue with thin collagen and fine elastic fibers. Only represents about a fifth or about 20% of the thickness of the total dermis. The surface area of the papillary layer is greatly increased by what we call dermal papillae, okay, or papillae, I don't know. I think it's papillae, the technical correct pronunciation. But these are small finger-like or nipple-shaped structures that project into the undersurface of the epidermis to increase the surface area. And there's that phrase surface area that I've referred to a few different times. Looking at this picture on the right, you really get a good view of some of those. Now it's labeled here as papilla. That would be one, since only one is labeled. But these are the projections of the dermis outward, connecting to the epidermis. Notice that it's a wavy connection between the two layers, the epidermis and dermis. In other words, just don't sit flat on top of each other. Like if you had two pieces of paper just sitting on one another, okay? What this enables is basically, like locking your fingers in place, or think of it like a puzzle, a jigsaw puzzle, where they contour to each other, and it gives great strength and connection to the two layers, so they're not, pulling apart. Okay, looking at the image here on the left that you saw on the first page, those little projections of the epidermis that you can see over here in kind of the dark red are projecting into the skin, deep into the skin. Those are called epidermal pegs. Okay, they match the contours of the dermal papillae. Okay, so that's how they lock in, and that's why the image kind of shown the way it is with those two separated. We'll talk a little bit more about epidermal pegs here on the next slide. Okay. Now, all of those dermal papillae will have capillary loops, which are blood vessels. Some will also contain tactile receptors called Meissner corpuscles, and others will contain nerve endings sensitive to touch. So if you can see here, like these little spaces, most of them, this one's kind of an exception in the picture, have something in there, okay? A nerve ending, a blood vessel, another type of nerve ending, okay? So that's where you would find some important stuff, okay? So still other papillae or papillae contain nerve endings, dendrites from neurons that give rise to sensations of warmth, coolness, pain, tickling, and itching, okay? Okay, the thicker layer is the reticular layer, makes up the other 80% of the dermis thickness. Consists of dense irregular connective tissue. We talked about that the other day. It means it's got those collagen fibers running in all sorts of different directions. It's not just collagen here. There are coarse elastic fibers, and then lots and lots of fibroblasts, those cells that I've mentioned. We do also find a few adipose cells. There's also hair follicles, nerves, sebaceous oil glands, sudoriferous or sweat glands that occupy the spaces between those collagen fibers. And I've already mentioned this, but the combination of collagen and elastic fibers in this region provides our skin with great strength, but also at the same time, extensibility and elasticity. Okay. Composition of the reticular layer gives the dermis the ability to attach to both skeletal and smooth muscle fibers, which in many cases are deep to our skin. And, you know, like in our face, when those muscles contract, like when we want to smile, the skin gets pulled along with it. Okay. In most places, the connection between the skin and underlying structures like our muscle is thanks to our subcutaneous layer or hypodermis, which, and you can see I've capitalized and underlined, it should not be mistaken as part of the skin, but it does connect excuse me, the skin and the underlying structures, which once again are primarily muscle. It is composed of areolar connective tissue, but most of it is adipose tissue, fat tissue. Okay, and this fat is going to help insulate you, so it helps with preventing heat loss. We also find nerve endings called lamellated or proscenium corpuscles that are sensitive to pressure here. One key feature of this layer that's really neat is it's highly vascularized. And the word vascular just means it's got lots of blood flowing through it. So it's got a lot of blood vessels, making it ideal for absorbing some drugs. Okay. So in some cases, like when a person gets a shot, they might inject that shot right into the subcutaneous layer. In fact, some needles are called hypodermic needles because more than likely that's where that injection's going. Now, if you get like the flu shot or the COVID shot, they're going to inject that into the muscle in your upper arm called the deltoid. That would be what we call an intramuscular shot, not a skin shot. All right. Any questions on this slide? I'm not going to go through and highlight the same things I already told you. And these two images, although really cool, are not going to be something I'm going to ask you to know for the exam. You guys fill in all the blanks. There's just 3 at the bottom, right?
Yeah.
Okay. Go ahead and get those filled out. Then we'll just go through and talk about everything. All right, at the top of the page here, we're going to talk about what we call skin markings. There's like unique features that skin will have. Some of them are more unique from person to person. We all will experience what we call striae or stretch marks developing on our body, hopefully not a whole bunch, but these occur when the skin becomes stretched too much, resulting in small tears of the dermis. Initially, they appear red, but become silvery white in appearance as they kind of become more like scar tissue. Very common in people that, stretch their skin in a short amount of time. So like if they put on a lot of weight or if, like a muscle grows. So like when somebody bodybuilds or lifts a lot of weights. Also very common in pregnant females because their belly has to stretch tremendously. And everybody's seen a very pregnant female, and the skin has to stretch to accommodate that. Okay, cleavage lines or tension lines, and the illustration here on the left side is going to illustrate what those are. In certain regions of the body, the collagen fibers within that reticular region of the dermis that we just talked about tend to orient more in one direction than in another because of the natural tension experienced by these regions of the skin, resulting from bony projections, orientation of muscles, and movement of joints. So I told you guys just a second ago that those collagen fibers in that deep part of our dermis kind of run all sorts of different directions. They do orient themselves kind of in a specific manner in order to provide good strength to the skin in ways that the skin's going to get pulled on. Most of the time, it just matches the way that the muscles of the body run, because when those muscles contract, they're going to pull the skin. So you want the skin to be strongest in those different directions. Now, why this is important? It's not something you can see from person to person. It's not really a super important thing for a lot of doctors or just normal people, but it would be especially important to plastic surgeons, because one thing a plastic surgeon wants to avoid is the formation of what? Scars. Okay. So looking at these two illustrations on the left, we show two incisions with sutures, one that has the incision going perpendicular to the flexure lines, or excuse me, the cleavage lines. The other is running parallel to it. Notice that figure A is showing you that suture is not quite as nice and tight, and thus that would result in the formation of more of a scar than what you see here. That's really nice and closed, less likely to split open, so would be much less of a scar, if one at all. Now, if somebody walked up and stabbed you with a knife, you can't control that, but a plastic surgeon definitely can control which direction they're going to cut. Okay. Epidermal ridges are seen in the surface of your palms, the fingers, the soles, and your toes, due to a series of ridges and grooves appearing as either straight lines or as a pattern of loops and whorls. So looking at your hands, you can see those little lines running all over the place. Okay, you see them in the palms and in your fingers. We form them during fetal development, so even before we're born. Okay, and these relate to those epidermal pegs that I was just talking about, extensions of the epidermis projecting into the dermis between the dermal papillae. Okay, they function to increase the surface area of the epidermis and thus increase the grip of the hand or foot by increasing friction. So once again, it's that jigsaw connection so that there's a really strong connection between the two. Okay. Because the ducts of sweat glands open on the tops of epidermal ridges as sweat pores, sweat and ridges are going to form our fingerprints or footprints on touching objects or when touching objects. So you have all heard of fingerprints, and now you know why we have them. Okay. Flexure lines or dermal folds typically are found at or near joints. If you use your hand as an example, you flip it over, you can see like where your knuckles are, that extra skin, those wrinkles of our skin, those are flexure lines or dermal folds. Okay. And the function of them is because, or they function to allow your skin to move when our joints move. Our skin cannot slide easily to accommodate joint movement. So in these places where we have joints, we have extra dermis tissue, and those folds and deep skin creases result. Okay, you've probably heard the phrase "skin tight." Okay, like if somebody's putting on a jacket or something, they say it's skin tight. It's really, really tight, and their arms and their torso really can't move very well. Our skin technically is not even skin tight. Okay, we've got these extra little bits of skin to enable our motion to occur. Okay, let's talk a little bit more about skin coloration, and that all begins with melanin that we've introduced a few slides ago, the pigment that gives not only color to our skin but also our hair, produced by melanocytes, stimulated by UV radiation. And I told you a couple slides ago, it's either kind of a reddish yellow pigment or a kind of brownish black color. And that's because there's two types of melanin, eumelanin and pheomelanin. Eumelanin is the darker brown colored pigment, whereas the pheomelanin is more reddish yellow. Okay. The major factor that affects your melanin production is your heredity. So in other words, your genetics, your parents control what color your skin is more than likely going to be. That's a major factor. Okay. It affects the type, but also how much. Okay, so this controls whether you are going to tan or not. Even if you're the most fair complected person and you don't tan very well, you still definitely do produce melanin. Probably just don't produce the type that you'd like and the amount that you'd like. That is the case unless you have one of the conditions I'm going to talk about in one second. Okay, another major factor in the amount of melanin we produce is our lifestyle. Okay, I've been talking about like tanning beds and spending time outside and being exposed to UV radiation. Well, if you're inside all the time, you're probably not going to be as tan as somebody that lifeguards in the summertime, right? Okay, so that affects your skin color. We also have variations that can occur to your skin color for a variety of reasons. Blood traveling through your blood vessels that are in your skin can affect your skin color. Now, people that are more dark complected, you won't see these conditions quite as much, but in people that are very fair skinned, you can see these changes tremendously. First condition, called cyanosis, is the blueness that occurs from a deficiency of oxygen in the cutaneous circulation. Typically is not a good thing at all. If somebody's deficient in oxygen in their blood, that's a very dangerous situation. I'll show you a couple of pictures here in the next couple of slides of these different things. Erythema is a redness due to dilated cutaneous blood vessels. So in other words, the skin is loaded with blood flowing through it. People that show redness, are experiencing like what? What can cause your skin to appear red? If you rub it, if you're embarrassed, or if you get mad. I mean, they say you get red faced because you're mad, or skin, when you get sunburned. Okay. Now, when when that sunburn happens, your skin hasn't turned red. It's because your skin has become damaged. And when our body gets damaged, whether it's the skin, a bone, a joint, whatever, we provide it with blood flow to try to fix that, because the blood is going to bring the stuff that can repair it. So your skin becomes loaded with blood flow, and that's why it appears that wonderful red color. And you all experience that, I'm sure. Jaundice is a yellowing of the skin due to an excess of bilirubin, which is a product of the breakdown of hemoglobin in our blood. Very common in people that are experiencing what organ failure? Liver. Okay. So like habitual alcoholics that have destroyed their liver will a lot of times experience jaundice. It's very common in babies, very newborn babies. Can be treated with black lights, which is actually kind of cool. But it's pretty wild how yellow an individual, if you've never seen somebody that has jaundice, even their eyes, the whites of their eyes can be really yellow. And I've got a picture of that here in a couple slides. Pallor is the pale color from a lack of blood flow. So in other words, the person kind of looks white. They say white as a ghost. And generally, if somebody like in their face looks white, what's going to happen to them? They're going to faint. Okay. Fancy term for a bruise is a hematoma. This is where you can see an actual blood clot through the skin. Like I said, most of the time, these conditions can be seen in more fair complected people. If you're darker skin, it's kind of harder to see these things. Okay. Bronzing is the golden color, golden brown color due to a condition called Addison's disease, where the adrenal cortex is low functioning. It's not like the bronzer that comes in a tube that you can put on your skin. That's not bronzing as far as this class goes. Albinism is a genetic lack of melanin. So in this case, that person will not produce melanin, and therefore their skin is really susceptible to that UV radiation. They look super, super fair complected. Their hair a lot of times is like sheet white. It's a genetic situation. Vitiligo is a partial or complete loss of melanocytes from patches of the skin, causing irregular white spots. You see this a lot in Black individuals. I got a couple pictures here in the next couple slides where you can see like their throat, very common, like the arms, where it just looks like their skin has been bleached in those areas. Carotene is a yellow-orange pigment found in egg yolks and carrots that can lead to yellowing of the skin. If someone, and this is gonna be like a really fair complected individual, eats a lot of egg yolks or carrots, they can actually look a little bit orangish yellowy, which is kind of wild. Tattooing, very common practice, permanent coloration of the skin in which a foreign pigment is deposited with a needle into the dermis. The key with a tattoo is it has to be involving the dermis. If you're just putting that ink into the epidermis, it's not going to be permanent, because what do we know? We're constantly shedding our epidermis. Okay. Moles, fancy term for those are a nevus, round, flat, or raised area representing a benign, that means non-cancerous, localized overgrowth of melanocytes that usually develop in Similar to those are liver or age spots, flat blemishes ranging in color from light brown to black due to accumulations of melanin. Darker than freckles though, and they build up over time due to sunlight exposure. Has nothing to do with liver. I don't know why that term is what it is. I guess it's just probably more than likely an old wives' tale would be my guess as to why they call them liver spots. Okay, like I said, I got a couple slides here to show you some pictures. Pretty good example of cyanosis, that blueness, very evident here in the fingers. Once again, from a lack of oxygen. Very common to see it in a person's lips, kind of almost purpley color to it. This is actually a picture of my oldest baby. He's not a baby anymore. Can you guys see like the left side of his face and down through his body, how it's redder than the right side of the body? Okay. This is showing you a kind of rare situation. If I remember, it's called Harlequin color change syndrome. Nothing was wrong with him, but for the first couple of days after he was born, it would just kind of randomly happen where half of his body would turn red. So he was showing erythema on one half. It was almost like all the blood just like moved to that side of his body. And if you kind of flipped him, it would actually almost like it was leaking to the other side. When it happened, this was actually when we had our like first checkup, like a couple of days after he was born. The doctor that delivered him and was checking on him, she'd never seen this before. It was really cool. Nothing, wrong with him or anything like that, but it's kind of a cool thing to see. All right, here's showing you that yellowing from jaundice. Like I said, pretty crazy how yellow the eyes can get, comparing the skin, somebody with jaundice versus somebody without, pretty evident. Albinism, once again, genetic lack of melanin. This lady would be an African American. Actually, she's a model in Africa, so I guess she would call her African American, but she has no melanin because of this genetic inability to produce it. So she's very fair complected. Here's kind of a couple pictures showing you vitiligo, where we are seeing losses of that dark complexion in the fingers, in the arms and legs. Can be very, tough on the individual. You start to lose, that normal skin coloration, and can lead to a lot of, emotional and tough things to think about. All right, keratinosis. This is where you can see that orange reddish tint to the skin because of, like getting too much of that keratin from an egg yolk or a carrot or something like that. You can see it down here in the hands. So kind of cool. I didn't include those in your handout, but I wanted to show you some clinical pictures anyway. All right, let's move on from our skin. Our first accessory structure we're going to discuss will be hair. Something that's pretty darn complicated. A lot goes on with our hair. Okay, fancy term for hair is pili. Now, pili is plural, pilus would be singular. You'll just hear me call it hair. It's a slender filament composed of columns of dead keratinized cells. So it's kind of like what we just talked about with the skin, and that there are these cells that have keratin and they're dead. They're bonded together by extracellular proteins. What's unique about your hair compared to your skin, it's actually made up of a harder form of keratin, more durable than the soft keratin that is found in your skin. Okay. Another key feature is that the individual cells are not going to flake off like they do in our skin. We find hair present on most surfaces of our skin, except places like the palms, palmar surfaces of our fingers, like the front of your hand and fingers, the soles, bottom of your feet, and the plantar surfaces of the feet. Okay. Main function of your hair is for sensory detection, but other functions also exist. Associated with each and every one of our hairs is a nerve ending called a hair root plexus. And even though your hair on the outside of your body is not living, when you touch it or when somebody touches it, you can feel that sensation. And that's one of the most important things that our hair does for us. Okay. Now, there are other functions of hair that exist. The hair that's on top of your head guards against physical trauma, heat loss, and sunlight. That's why, like my dad, who's bald, has to wear a hat. We'll put a lot of sunscreen on there. Eyelashes and eyebrows help shield your eyes from foreign particles. Your nose and your ear hairs also protect from foreign particles. I don't know why, as guys get older, we need more and more nose and ear hairs, but we do apparently. So those are kind of just the general functions of hair. You could also throw in there, cosmetic, which, can lead to like a physical attraction between the opposite sex or the same sex, which can lead to reproduction. And I won't get into that. Structurally, hair consists of kind of two major regions, a shaft and a root. The shaft is a superficial portion that's above the skin surface. So that's the part of your hair that we can see. That's the part of your hair that you can get cut. If you keep your hair long, you keep the shaft of your hair long. The root is like the root of a tree. It's embedded in the skin. So in other words, it's not visible. It's inside, and that's really not going to be any different in size, whether your hair is really short or it's really, really long. Okay. Both the shaft and the root of our hair consist of 3 concentric layers of cells, and we can see those here in our picture on kind of the bottom here, letter C. Now, the hair root is just shown here in the bracket because surrounding it, we'll talk about what the hair follicle is here in a second. But you can see these 3 kind of ringed layers, okay? And kind of this purple layer, the outer layer, is what we call the cuticle, the surface layer of scaly cells overlapping each other like roof shingles with their edges facing upward. In the middle, the very middle part in yellow here would be what we call the medulla, core of loosely arranged cells and hair spaces. It's found in thicker hairs, but not going to be found in thin ones. Okay. And between the medulla and the cuticle is the cortex. That's the layer of keratinized cuboidal cells. Okay. That would be what's shown here. What is this, kind of a reddish pink, peach color? Okay. Now, that's important to understand, but if you've ever wondered why your hair has a specific texture, whether it's straight or curly or wavy, it relates to this shape. In other words, it's cross-sectional shape. If it's nice and round, it's going to cause your hair to be more straight. Okay. If it's more oval shaped, your hair will be more wavy. If it's flattened, it's going to cause your hair to be more curly. And that, just like hair color, a lot of it's determined genetically. Okay, now you can do different things to affect that, but your traditional normal hair texture relates to your cross-sectional shape. Speaking of hair color, it's due to pigment granules in the cells of the hair cortex. Okay, we just talked about eumelanin, pheomelanin, same things here. The same pigments are found in our hair. If your hair is brown or black, it's because you have lots of eumelanin, and people that are typically dark complected generally have darker hair. Red hair has a little bit of eumelanin, but high concentrations of pheomelanin. Blonde hair has an intermediate amount of pheomelanin, but very little eumelanin. And those people that, as we get older, become more gray and white haired, it's because it's absence of nearly all melanins and contains air in the medulla. So it's not as healthy of a hair, which, you can kind of see that just by looking at it. Okay, as I mentioned, we're going to talk about the structure that encloses the hair root, what we call the hair follicle. Okay, you can see it's labeled here in this picture where we've kind of exposed different layers. Let's do that now. Hair follicles are oblique tubes in our skin that surround the root of the hair. So it's basically just a structure or a tube that encloses the hair and holds it in place. There's 2 parts to it, an external and an internal root sheath. Collectively, those are what we call the epithelial root sheath. Okay. looking at our image here, you can see internal root sheath, external root sheath. Okay, those are also labeled down here at the bottom, although I'm not really worried about you identifying those kind of other pictures. The dense dermis that surrounds the hair follicle is called a dermal root sheath. So in other words, the thick skin around that, it's not labeled, would be the dermal root sheath. And that just kind of is the name of it. Okay, other things I want to talk about. The bulb is an onion-shaped structure found at the base of each hair follicle, which is very visible here in our figure A. It's just the fattened kind of onion-shaped or rounded shaped segment of our hair follicle at its base. At the very innermost part of our hair follicle is the bulb. Okay, now if I zoom in, you can see this is where you're going to find surrounding it a structure called the hair root plexus. That's that nerve ending that I was talking about that enables us when somebody touches our hair shaft or when we touch our hair shaft to feel it, to sense that. Okay, at the tip of the hair bulb is a little indentation called the papilla of the hair or hair papilla. You can see it's a little indentation. We see two blood vessels associated with it. Okay, these blood vessels are going to bring nutrients and remove wastes away from our hair. Internally, so all the way, as far inside in our hair root, we have living cells, just like our skin. Okay, our hair grows from the inside out. Okay. Once that hair is pushed out of the skin, and that's the hair that we can see, that's the hair that we can cut, that's all dead cells. But these cells here in what we call the hair matrix are living, and they are the cells that are responsible for going through cell division to produce the hair cells that eventually get pushed outward and eventually die. This is also where you're going to find the melanocytes, those same melanocytes like we had on our skin, that provide the pigmentation, so the control of our hair color. Okay. Now you've probably heard that if you cut your hair, it's going to grow faster and stuff like that. Best way to make your hair grow faster is make sure you get good nutrients, which we really can't control. Cutting the outside of your hair shaft, that's just dead tissue. That's not going to make it grow any faster. So that's probably one of those old wives' tales. We talked about these. All right, let's talk about some other structures associated with our hair. We're going to talk more about these in a little bit, but sebaceous glands, fancy term for oil glands, are associated with our hairs, and they secrete an oily substance directly into the hair follicle. This explains why if you go a couple days without washing your hair, it's going to kind of have that oily texture to it, because you're literally dumping oil onto your hair all the time. An erector pili muscle is associated with your dermal root sheath. This is also going to go by the term piloerector. It's going to do exactly what its name sounds like. Pili, we said, means hair, right? Erector means it like erects it, it makes it stand up. When you get cold or when you get frightened, your hair will stand up on end. Okay, it's not as noticeable in humans because we don't have a whole bunch of hair like an animal does, like my dogs. When they get frightened or upset, you can see their hair standing up on the back of their spine. Okay, we just see what we call goosebumps. Okay, the whole function of it in animals with fur is it makes them more intimidating. At least that's the thought process. But it also, when they stand up on end, the hairs become better insulators. So that's why it happens when we get cold. But since we don't have that much hair, it really doesn't do a whole lot for us. Okay. Now, if I go back to that very first page, okay, here it's listed as erector muscle of the hair. You might want to go to that first page and label it, put in parentheses or whatever, as erector pili muscle. So I want you to know it. I don't care about piloerector, but erector pili, make sure you know it that way as well. All right, there's several types of hair that exist. Lanugo hairs are very fine, non-pigmented downy hairs that cover the body of a fetus. So this is all interuterine life, so before the baby's born. They're basically found all over the body. A lot of those are even lost before the baby's born. Terminal hairs are long, coarse, heavily pigmented hairs that replace those lanugo hairs of the eyebrows, the scalp, and eyelashes prior to birth. Not all babies are born with hair on their head, but those that do, the hair is terminal hair. In the case of, like, you can see my baby a couple of slides ago, he had really dark hair on top of his head, and now his hair is like bleached white. Okay, so it's not always indicative of what the individual is going to have for the rest of their life. Vellus hairs are short, fine, pale hairs that are barely visible to the naked eye, replacing lanugo hairs over the rest of the body. You look at like your face, for example. Even if you're a lady without a beard like myself, we have these tiny little vellus hairs. Okay, commonly called these peach fuzz. And in response to hormones secreted at puberty, those hairs eventually become replaced by those terminal hairs, much darker, thicker hairs. During adulthood, about 95% of the hair on a male's body is terminal, and then only about 5% are those peach fuzz vellus hairs. Okay, men are much, much hairier than females, and that's pretty common sense stuff. About 35% of the body hair of a female is terminal, and that means about 65%. So the majority of her hair will be that peach fuzz vellus hair. Looking at this, that just same figure, basically just showing you what's over here. These two images, I'm not going to worry. I did reference them a little bit, but this is the image that I want you to know for the exam. I would say everything in this picture would be fair game, with the exception, don't worry about blood vessels, so you can cross that off. Now, we're going to talk more about apocrine sweat glands, which are visible here in this picture and are associated with our hair, but we'll do that here in just a second. Here we go with another one of those clinical pictures, kind of showing you zoomed in 70 times, which your naked eye can see. Here's the skin surface where you can see those flaking dead skin cells, and you can see the hair shaft extending out. You can see the overlapping cells, how they are facing upward, which is kind of cool. Not something you don't need to worry about. Here we go with a couple of pictures showing you a newborn baby with those tiny little non-pigmented downy hairs. You can see an example of some goosebumps where the hair is standing up on end. That kind of peach fuzz type hair called vellus hair. Okay, moving on from hair, our next accessory structures we're going to talk about will be your glands. Thinking back to Tuesday, I kind of introduced glands and glandular epithelium. We learned in that lecture, a gland can be described as a cell, group of cells, or an organ secreting substances, either for use elsewhere in the body, like in the case of hormones, or just to be released from the body altogether, like in the case of sweat and oil. Okay. There's two types of glands, and we talked a little bit about these the other day. Endocrine glands have a high density of blood capillaries and secrete their products directly into the blood. Those products are hormones. We saw the picture of the thyroid gland, and I told you guys we'd talk about endocrine glands much later in the semester. Associated with our skin, and that's why we're talking about them in this lecture, are exocrine glands. They maintain their contact with the surface via a duct, which is just an epithelial tube conveying their secretion to the surface. Okay, that's what we can see here kind of in our picture. Nice duct carries it from where the secretion takes place, in the case of our skin, to the surface of the skin. But not all exocrine glands are associated with the skin. Your salivary glands, Any of the digestive glands in like the stomach or intestines, they are also exocrine glands. Instead of connecting to the surface of the skin, they would be like, to the lumen of that organ inside of the mouth, inside the stomach, whatever. Okay. Now, most of our exocrine glands are enclosed in a fibrous capsule that divides it up into lobes and lobules, just smaller subunits. Cells that perform the task of synthesizing and secreting are collectively called the parenchyma of the organ. And these are kind of terms I don't refer to a whole lot in this class. There's a couple different ways to kind of categorize our various exocrine glands. We're going to look at the way that we kind of structurally classify them here. And then at the bottom, we're going to talk about how we functionally classify them. Okay. We classify them structurally according to two criteria: whether their ducts are branched or unbranched, and then the shape of the secretory portions of the glands. Okay. So looking at our illustrations here, we've got 5 and then 3 more. There's 8. We have some of these that are simple, some of them that are compound. Okay, all of these 5 top ones are simple, which means that they have a single unbranched duct. In other words, like this purple shaded area, the duct doesn't have a branch to it. Okay, whereas compound, the duct is branched. Okay, it's as simple as that. Okay, now kind of in the pink color, that's the secretory portion of the gland. Okay. If the duct and the secretory portion are of uniform diameter, so in other words, if it's about the same width here versus up here, we'd call it a tubular gland. So in this case, it's a simple tubular. Okay. If the secretory should be an O there, cells are more sac-like, we call this a cinar. And you can see it's a lot fatter here than it is up here where the duct is. If the gland has both secretory cells in the tubular and acinar portions, we'd call it a tubuloacinar. Okay, where it's basically got kind of a combination of both tubular and acinar. Okay. As far as these 8 pictures, I don't care about you knowing them. I just wanted to kind of introduce it. It's in your textbook. We don't really refer to glands that way most of the time. The main way that we describe glands is based on their functional classification, which relates to how their secretion is released. And these 3 images I actually do want you to know. The first type, what we call merocrine glands, they release their secretion by a process called exocytosis. So this is where, and it kind of relates to stuff we talked about earlier in this unit, where the RER, endoplasmic reticulum, makes like our proteins. The Golgi apparatus or Golgi complex is like the FedEx or UPS. It creates those vesicles, those little bubbles. Inside of the vesicles will be the secretion, and then that secretion is released when those vesicles just kind of fuse with the plasma membrane. Example of merocrine glands, you can see our salivary glands, and the ones also in our pancreas are going to operate this way. Okay, in an apocrine situation, it's kind of similar in that we create those bubbles, those vesicles from the Golgi, but instead of the individual vesicles being released one at a time, they accumulate on one side of the cell. And as you can see, the cell kind of pinches inward, and that part of the cell just breaks off or is pinched off. And so part of the cell becomes part of the secretion. Okay. In this case, an example, as you can see, would be mammary gland in a lactating female. And after the cell breaks into its little kind of smaller segment, it's just going to repair itself and repeat the process. And the final type are called holocrine glands. Very similar. We create those bubbles and they accumulate, except instead of part of the cell breaking free, just the entire cell just ruptures and becomes part of the secretory product. And so the cells are dying off, which means we constantly have to replace those old ones with new ones via cell division. And that's what's shown there at the bottom of the page. Example would be those oil or sebaceous glands that I've already kind of referenced. And like I said, I do want you to be able to identify the 3 different functional types of hormone, or it's going to be glands that are illustrated there on the right side. All right. Let's talk now about the major cutaneous or skin glands associated with the integumentary system. That includes our sudoriferous, kind of a wonky word, fancy term for our sweat glands. There's two types of those, eccrine and apocrine. Also going to talk about our sebaceous or oil glands, which I've already referenced. Our earwax glands have a fancier term, what we call ceruminous glands. And they secrete cerumen, hence the name. That's just a fancy word for your earwax. And then the milk producing glands in a pregnant and lactating female are the mammary glands, which I've already kind of talked a little bit about. Okay, let's start with sudoriferous glands. Once again, we commonly call these our sweat glands, most numerous glands of the skin. So sweat is going to be the biggest product released by our skin. They produce a water perspiration that serves primarily to cool the body. We all know sweat helps keep us cool, and that's why we do it when we get hot. We find sweat glands over the entire surface of the skin except the nipples and parts of the external genitalia. And two types of them exist, eccrine and apocrine. Okay, if you go back to that very first picture that I had you guys highlight things in, I had you highlight both of them. I'll tell you how to identify the difference between them in 1 second. Okay, eccrine are the primary sweat glands that are used to cool the body. I'd probably highlight that phrase. That's important. Abundant in places that sweat a lot: our palms, the soles, bottom of our feet, front of our hands, and our forehead. Okay, they are structurally going to consist of a simple coiled tubular gland where the secretory portion lies in the dermis. The duct opens to a funnel-shaped pore at the skin. Okay, sweat produced by them consists of water, which is most abundant, ions, mostly we're talking sodium chloride, which are the ions that are used to make salt, urea, which is a waste product, uric acid, a waste product, ammonia, another waste product, amino acids, glucose, and lactic acid. Okay. Now, apocrine sweat glands are simple coiled tubular glands found mainly in the skin of the armpit, the groin, areolae, surrounding the nipples, and the bearded region of a male face. Okay. The secretory portion of these glands is located mostly in the subcutaneous layer, and the excretory duct opens into hair follicles. So here's the difference. Apocrine, are always associated with a hair follicle, which if I go back to that very first page, that very first picture, if you look where we can see the coiled portion of the gland, that's not really what's important. Just follow the tube, the duct. If it leads all the way to the surface of the skin at what we call a sweat pore, you know it has to be an eccrine sweat gland. If that duct leads to, and you can see it coming in right here, if it leads to a hair follicle, it has to be an apocrine. So in other words, apocrine sweat glands aren't going to be found in places where there's not hair, okay? Because they're associated with hair. So that's why you find them in the armpits, which, you know, are hairy, the groin, which is hairy, and so on and so forth. Obviously, the beard is not a hairy place. Now, the sweat that they produce is a little bit more unique. It contains fatty acids, which bacteria love. Bacteria, and I'm going to say this kind of loosely, they kind of eat it, and that's not a real way that they do it, but they utilize it to get energy. And in doing so, they give off gaseous odor, and that's going to produce our body odor. A fancy term for that is called bromhidrosis. Okay. Okay, sebaceous or oil glands are simple branch center glands where the secreting portion of them lies in the dermis, usually opens into the neck of a hair follicle. We kind of talked about that already. That explains why your hair becomes oily. Okay, we find them all over the body except the palms and the soles, which are places that we don't want to be oily, because those are the places that we want to, have friction, and oil would reduce friction because friction is good for like gripping things, but also because we don't have hair on our palms and soles. Okay, and these are glands associated with our hair. They produce an oily secretion called sebum, just a fancy word for oil, a mixture of triglycerides, which is a form of fats, cholesterol, proteins, and inorganic salts. The function of sebum is to keep your skin and hair from becoming dry, brittle, and cracked. And very common for this to become inflamed or infected and lead to what's called acne, which we've all experienced, commonly called pimples and zits. Ceruminous glands are modified sweat glands specific to the external ear. Their secretion combines with sebum or oil and dead epidermal cells that form cerumen, fancy word for earwax. Function of that is to provide a sticky barrier to foreign bodies and insects, keeps the eardrum pliable so it doesn't become dry and cracked, waterproofs the auditory canal, and can actually help kill bacteria. That's what that phrase bactericidal effect means. The secretory portion of these guys lies in the subcutaneous layer, and their excretory ducts open either directly onto the ear canal, or I should say into the ear canal, or into ducts of sebaceous glands. We'll talk more about them, and we'll see a picture of them when we talk about the ear. Well, I guess we won't see a picture of them, but that's okay. Mammary glands are milk-producing glands developing within the female breast only during pregnancy and lactation. They're modified apocrine sweat glands that produce a much richer secretion and channel it through ducts to a nipple for very efficient secretion. Very last slide of the semester that we'll look at, we'll talk more about mammary glands, and that'll be in our reproductive lecture. All right, and then one final slide. Our final structures to discuss will be your finger and your toenails. All right, we can describe nails as scale-like modifications of the epidermis that form a clear protective covering on the dorsal or posterior surface of a finger or toe. Each is composed of hard keratinized epidermal cells. They function to protect the distal end of our digits, so the very ends of our fingers, enhance touch perception and manipulation, allow us to grasp small objects. And one of the best things they can be used for is just to scratch and groom the body in a variety of ways. Okay. Structurally, they include quite a bit of things. The nail body or plate is the visible portion of the nail, comparable to the stratum corneum of the epidermis, with the exception that its flattened keratinized cells are filled with a harder type of keratin, and the cells are not going to be shed off. Okay, so that's what the nail body also goes by the term nail plate. Okay. Extending from that is the free edge. That's the part of the nail that will be of variable length, because this is what you would cut. Okay, it's white in appearance because there are no underlying blood vessels called capillaries. Okay, so that's at the distal end. At the proximal end, back towards where the nail begins, there's another kind of whitish area. We call that the lunula, a whitish crescent-shaped area of the proximal end of the nail body. It appears white due to a thickened region of epithelium in that area. Okay. It gets its name, I believe, because it kind of looks like a quarter of a moon. The word lunar refers to moon, and so that's where that term lunula comes from. Just like our hair, there's what we call our root. In this case, it's the nail root. That is the portion of the nail buried in a fold of skin, and you can see that very nicely. A fun word to say is the next structure, what we call the hyponychium, a thickened region of the corneum beneath the free edge. This is what helps secure the nail to the fingertip, as you can see there. The nail bed is the skin below the nail plate, extends from the lunula to the hyponychium, shown here in our picture. And then the nail matrix is a lot like the hair matrix. It's at the proximal end where the nail root is. This is where cells are going to go through mitosis, cell division, producing new nail cells, which enables the nail to grow in length. And then another fun word is the eponychium, which is most commonly called the cuticle. A narrow band of epidermis extends from and adheres to the lateral border of the nail wall, occupies the proximal border of the nail, and consists of stratum corneum. So that's that little kind of flap of skin. We commonly call that eponychium or cuticle, but it's fancily known as the eponychium. I do want you to know these two pictures. I would say everything in this picture we talked about, so highlight that. And this picture, just worry about the structures that are capitalized for you. So don't worry about epidermis, dermis, phalanx, which is just a fancy term for the finger bone. And then looking at the picture at the bottom, it's showing you a photomicrograph, so special scan of a fingertip from a cadaver that's been cut, just like this one. Question?
Yeah, what are the little white spots you get on your nail as it grows out?
I've heard that's calcium deposit. I don't really know. Like, so the question, if you didn't hear, was like, what are the little white specks that you kind of see sometimes? I'll see if I can research it, answer that question, because I don't know off the top of my head. Very, very good question.